Hi. Thanks, Adriana, for inviting me. So I will present our recent fMRI study conducted with my colleague Sébastien Hux from McGill University across Sébastien Jacquemont Lab at Saint-Justine Montreal Hospital, Pierre Bellec Lab at the EUGM Research Center, and Kerry Burden Lab at UCLA. So we investigated shared functional connectivity alteration across neurodevelopmental mutation, autism, and schizophrenia. So neuropsychiatric disorders have mainly been studied using a classical top-down approach, starting from the psychiatric diagnosis and moving down to cognitive dimension, brain endophenotype, and genetic factors. So the DSM-first approach has attempted to characterize stable endophenotype signature for each clinical condition independently. Such case control framework has led to mixed findings with theory of over as well as under connectivity in the example of autism, and highlighted a genetic overlap between conditions, between ASD, schizophrenia, and other neurodevelopmental conditions, leading researchers to conduct cross-disorder analysis and to leave this typical framework. So this strategy revealed shared signature across diagnoses. So for example, MRI study reported a gray matter loss converging in dorsal anterior cingulate cortex and insula. An fMRI study recently highlighted shared networks across ASD, schizophrenia, and ADHD, in particular in the salient, frontoparietal, and DMN network. And at the genetic level, the Psychiatric Genomic Consortium performed the meta-analysis of the 8 largest GWAS, and they reported as well more than 100 loci associated with at least 2 psychiatric conditions. So to conclude, and as many of you may have noticed, our gene don't seem to have read carefully the DSM. So a complementary gene-first approach has recently been used to dissect neurodevelopmental disorder, starting with identifying and building models from a lower level in the hierarchy, and then asking questions about how such low-level models can explain phenomena higher up in the hierarchy. But to use the bottom-up approach, studies need to focus on genetic variants with a large enough effect size and which are relatively frequent to assemble a sufficiently powered cohort. So we used for that copy number variant, which are deletion or duplication of specific genomic region, and they have been the most appropriate mutation to conduct such studies. They are characterized by mirror gene dosage effect on specific traits such as BMI, head circumference, and even the diagnosis. So for example, we studied the 16p1.2 deletion, which have mainly been associated with autism, macrocephaly, and obesity, while duplication have been associated with schizophrenia, microcephaly, and anorexia. A mirror gene dosage effect has been as well reported in the insula volume. And in an opposite way, the 22q deletion syndrome has been shown as the highest known genetic risk factors for schizophrenia, while the duplication of the same genomic region has been mainly associated with autism and ADHD. However, no fMRI study have been conducted to date at the gene to investigate the mirror gene dosage effect. So it's why we first attempt to characterize the functional connectivity profile of those recurrent CNVs with the hypothesis of a mirror gene dosage effect. So for this purpose, we used 2 CNV first cohorts provided by the Simons VIP Consortium and UCLA through Kerry Burden Lab collaboration. And we used as well an unselected population as a normative model from the Brain Genomic Superstruct project. And with idiopathic samples that I will describe later, and after fMRI pre-processing with the NIAC software developed at Montreal by Pierre Bellec and standard QC procedure, we ended up with 3,500 connectomes. So we performed a parcellation into 64 functional seed regions, and functional connectivity was estimated as a temporal pairwise Pearson correlation between the average time series of these 64 seed regions. We obtained 2080 correlation coefficients that we converted to standard scores. All the following statistical models were performed at the level of these 2080 pairwise functional connections estimated between these 64 regions. So we first asked if we were able to report the non-clinical gene dosage effect of CNV at the whole brain level. And the answer was yes. So for the 16P in the left panel, we observe an overconnectivity for the deletion group compared to control, which is in the y-axis, while the duplication exhibits an underconnectivity in the x-axis. So as expected, we found an opposite pattern for the 22q in the right part, with underconnectivity associated with deletion and overconnectivity with duplication. And finally, it was interesting to observe that this global shift is going in the same direction for the 16p duplication and the 22q deletion, which are the two CNV associated with schizophrenia. This global underconnectivity shift has been previously reported in case control study in idiopathic schizophrenia. So then we ask whether this CNV alteration could be related to the non-deletion effect on head circumference and brain volume. But however, the relationship between brain volume and functional connectivity is not yet clearly established. So we perform linear regression in the large unselected population, so the GSP cohort, to investigate the linear effect of total brain volume on the 2080 functional connection. So we show an effect of brain volume and functional connectivity, which is reported in green in the x-axis, but this effect was 10 times smaller than the one associated with both deletion in the y-axis. So it is therefore likely that brain volume could mediate at least a small part of the effect of the deletion on functional connectivity, but we cannot say that brain volume alone explains our reported signal of CNV on functional connectivity. However, further analysis need to investigate the relationship between total brain volume and functional connectivity carefully. Finally, we ask if specific networks were altered and responsible of the general shift observed in the 16p deletion group. So among the 2080 beta value corresponding to the 2080 connection, the FDR significant one in red were grouped into their respective 64 seed regions, the gray inner circle, and then their respective 12 functional networks, where color labels are reported in the legend below. So they are displayed in this core diagram with red color corresponding to overconnectivity. And we reported that our overconnectivity shift in 16p was mainly driven by the ventral attention network and motor network. We did the same for the 22q deletion group, and you can observe in this plot in the right panel that even if we have a general underconnectivity profile in blue, it's not everywhere, and it was driven by the limbic, frontoparietal, and anterior DMN, for example, in pink. So it's finally interesting to note that we reported a known higher connectivity, known in schizophrenia, between thalamic region and auditory network. We then ask if functional connectivity signature of both deletion may represent dimension contributing to idiopathic autism, schizophrenia, and ADHD. In other words, can this connectivity signature help to dissect this multidimensionality nature of psychiatric condition? So we hypothesize that because deletion are overrepresented in autism, schizophrenia, their functional connectivity signature may provide insight into dimension contributing to this condition. So to test that, we first characterize the functional connectivity signature of the 3 independent idiopathic cohorts that we compare to our deletion functional signature defined by gene-first bottom-up approach. A quick note to point out that we refrain from investigating the effect of duplication at the individual connection level due to the smaller sample and effect size. So we performed 3 independent contrasts between patients and their respective control, correcting for age, sex, motion, and sight, and we compare them with 2 contrasts of the deletion group. The density plot represents the distribution of the 2080 beta value for each contrast. On a note, each connectome has been previously standardized based on the variance of the respective control group. So we reported at the whole brain level an underconnectivity profile for schizophrenia and ISD, which was stronger in schizophrenia, reported in yellow, but however, effect size observed for both deletion were 2-fold higher. Finally, we correlated the 5 functional connectivity profile against each other, and what we see in the right part is that the highest correlation was between ISD and schizophrenia, and surprisingly between 22q and ISD, as well as 16p and schizophrenia. We did not expect to have such correlation at the connectome-wide level because we were assuming that only a restricted set of connections would overlap between deletion signature and this idiopathic profile. So to dive into that deeper, we investigated individual seed region profile driving this correlation. For this purpose, we tested a functional connectivity profile computed for each subject with or without a diagnostic of either ASD, schizophrenia, or ADHD, could exhibit similarity to either 16p or 22q functional connectivity signature. This was performed for each seed independently. So we computed the similarity score for each individual functional connectivity profile with the 6 deletion functional connectivity signature. We then compared similarity score between case and control for each seed region independently, and we reported seed region with significant group difference after SBR correction. So, for the example of the putamen here, data point in the box plot represents the similarity score of each individual with autism and each control subject. So we reported similarity of individual functional connectivity profile with both deletion functional connectivity signature. So, for example, functional connectivity with the 16p deletion signature was significantly higher in individuals with ASD compared to control for 6 out of the 64 seed-based functional connectivity patterns, which are reported in the pink ellipse. For example, here the putamen contribute to the similarity between individuals with autism and the functional connectivity signature defined by the 16p deletion. For this region and for the thalamus, individuals with either schizophrenia, which are in the left panel, or ASD, in the right panel, show higher similarity with both deletion. None of the deletion functional connectivity patterns show higher spatial similarity with control. We furthermore carefully investigated that none of these results were related to motion, and it was not the case. And in the same way, we investigated that none of these results were related to motion. And it was not the case, I'm sorry. In the same way, we investigate that the relation with cognitive scores and found that the specific seed region enriched in autism that exhibit a higher similarity with both CNV signature, more they exhibit a higher similarity with CNV signature, lower was their IQ and higher was their ADOS measure, in particular in the caudate, anterior insula, and cingulate cortex. So overall, there is a broad similarity between deletion functional signature and ASD and schizophrenia. So this deletion points toward a functional connectivity dimension that may represent an important building block for some individual with psychiatric condition. So it's why we extended our work by computed connectivity similarity between this seed-based functional pattern and of both deletion profile, and we compare them to a neural model of correlation between random group contrast that was generated over 10,000 permutations in the unselected large cohort. So we reported a positive significant correlation between both deletion in the anterior DMN and frontoparietal network, driven more specifically by the dorsomedial prefrontal cortex and the anterior cingulate cortex. So these two functional networks, it's interesting to note that they have previously been reported in cross-disorder fMRI studies across ADHD, schizophrenia, and ASD. So to conclude, our current studies show a mirror gene dosage effect of 16p and 22q, a higher similarity of individuals with ASD and schizophrenia with both deletion signature, and for specific psychiatric associated seed region, and finally, a covariance profile between haploinsufficiency, functional connectivity signature, which means the deletion signature, which suggests that this altered dimension may represent building blocks common to several psychiatric conditions. So we aim to extend our approach to other neurodevelopmental CNVs with an hypothesis of shared molecular pathway affected across CNVs, corresponding to a limited number of latent variables reflecting different mechanisms like pleiotropy, and functional connectivity signature could be later mapped into large general population dataset to be associated with cognitive dimension. So thanks to all my colleagues, in particular Sébastien Hux, and to my co-supervisors Sébastien Jacquemont and Pierre Bellec at Montreal. And I would like to really thank all the consortium that share the data with us. and all the family participating in this large project at the Saint-Justine Montreal Pediatric Hospital.